Hello everyone. The topic of my research is antibacterial effectiveness of Citrullus lanitus mediated stannous nanoparticles on Streptococcus mutans. As we know, nanotechnology involves the manipulation of bulk materials to a range of something which is 1 to 100 nanometers. This means that you scale down the size, which gives more novel biological properties like optical properties, more catalytic properties, and hence makes nanoparticles easier to get absorbed and produce biological changes which we require. They can be metal based, carbon based, ceramic or polymer based, or even composites. The focus of my study is stannous nanoparticles in the form of stannous chloride. The synthesis of nanoparticles generally involves some kind of physical or chemical methods. The disadvantage of these are basically there is high cost, high energy expenditure, and the toxic byproducts which are released by nature are really susceptible to degrading the biological properties. To combat this, there is something called green synthesis, which came into existence, wherein you use plant extracts as stabilizing, reducing, or capping agents. This method is more cost-effective and non-toxic approach. Stannous nanoparticles in general have found to have antibacterial, antifungal, and photocatalytic property. That is, they are readily reducing reactions as a catalyst in the form when in the presence of for light. The biomolecules in the plant extracts, such as flavonoids, citrate, and phenols, basically are the active components which are responsible for capping, reducing, and stabilizing of nanoparticles from the parent cation. Citrullus lanitus, which is nothing but watermelon, extract has been found rich to be in antioxidants, phenols, and flavonoids, which thereby help the stabilization of such nanoparticles. Hence, fruit extract mediated nanoparticles are synthesized, which are much cost effective and have warranted more research into the chemical reduction mechanisms which are exhibited by these plants. The objective of my study is to synthesize and perform characterization of the watermelon mediated stannous chloride nanoparticles and to evaluate the antimicrobial efficacy against Streptococcus mutans for the test groups, which are basically controlled, which is distilled water, your stannous nanoparticles, and 2% chlorhexidine gluconate. The materials and methodology is such. The synthesis of plant extract was done by cutting watermelon fruit, which was of native variety, because it has high phenolic content, into smaller pieces and weighing them for 50 grams. And thereby crushing them in mortar and pestle to basically get a fruit extract. Now, this fruit extract was added with distilled water, 200 ml, and then using Whatman's filter paper, 50 ml of the filtrate was obtained. On the other hand, the nanoparticles, which was stannous chloride powder was measured for 0.45 grams, which was then diluted in 60 ml of distilled water. This 60 ml was added to the 100 ml for 250 ml of the filtrate, which was obtained from the plant extract, and thereby stannous chloride solution was obtained. The stannous chloride solution was subjected to orbital shaker so that we can ensure uniform nanoparticle density and size, because constant shaking is what produces the nanoparticles. This was then centrifuged at 8000 RPM for 10 minutes to produce a filtrate and a supernatant. The supernatant was given for characterization, and the filtrate was basically taken and obtained in the form of stannous nanoparticle pellet. In order to increase the bioactivity of the nanoparticles, the rind of the watermelon, which is super rich in antioxidants and antimicrobials, was cut, grind, diluted, and filtered. The filtrate was boiled to obtain a final concentration of 5 ml, which was then added to the nanoparticles which have already been synthesized. So 1 ml of synthesized nanoparticle plus 1 ml of the concentrated rind derived solution was together mixed in a vortex mixer, which was thereby obtained to carry on with the results. This solution was then plated, half of it was plated in the antimicrobial agar gel to form agar dilution gel against stannous mutants. And the other half was used to find MIC in a microbial broth. The results were as followed. 50, 100, and 150 microliter of the derived solution was plated in the agar diffusion plate against chlorhexidine 2% gluconate. This showed that there was some amount of minimum inhibitory concentration, which was found around the gel plate. Apart from that, they were filled in wells, which was basically read through the ELISA microplate reader, which basically determines the amount of MIC, which is in minimum inhibitory concentration. The minimum inhibitory concentration is the amount of antibiotic or antimicrobial agent which is needed to inhibit the amount of microbial activity which is present in that. The first three wells were filled with 50, 100, and 150 microliter concentration of the sample. Then a control was used in which there was no bacteria, no antimicrobial agent, but only bacterial growth was left uninhibited. And then chlorhexidine 2% gluconate was used. The readings were taken at 1, 2, 3, 4, and 5 hour intervals and have been presented in the table as follows. Apart from that, EDS, which is elemental system, and EDS analysis was basically done in the given sample as shown, in which it was seen that carbon was 48%, oxygen was 29%, stannous was 15%, and chloride was 4%. This shows that the elements which are present in the nanoparticles, which was stannous chloride, are present as follows, apart from the basic carbon and oxygen. Apart from this, FTIR analysis was done as a characterization procedure in which peaks were obtained at certain part using the infrared spectra of the stannous chloride sample. These basically showed absorption peaks ranging from 800 to 2400. To be more specific, there were double peaks which were ranging from 2339 to 2358, which is a strong for O double bond C stretching mode of carbon dioxide. The peak of 2005 is medium for C double bond C stretching mode in the allene group. The peak at 1011 is strong for C double bond F stretching mode of the fluoro compound, and the peak at 814 is basically strong for CH bending. These are functional groups of stannous fluoride particles and thereby show that the stannous fluoride nanoparticles are abundant. The discussion is as follows. Alkaloids and the free reducing sugars are present in all parts of watermelon. These act as reducing and capping agents for stabilizing the stannous cations to nanoparticles. Characterization of the nanoparticles is necessary to understand the chemical and physical properties of the elements involved. Since bioactive properties depend on the element involved, characterization of the sample helps in appreciating the bioactive compounds. The preliminary results show that watermelon-mediated stannous nanoparticles are stable in nature and they show promising results for the future in terms of antimicrobial activity. The antimicrobial activity and efficacy has to be further developed and explored in future studies. Thank you.